How do citizen science projects build community and drive new areas of research? What roles do metabolites play at the interface of microbiome and host? And what is a healthy vaginal microbiome made of? These are just a few of the questions we answer in this episode of The Metabolomist with my guest, Caroline Dricot. Welcome to Season 5 of the Metabolomist podcast, where we dive into the communities that are transformed by metabolomics. I am your host, Alice Limonciel, and this year I will engage with scientists who not only use metabolomics, but who leverage it for the greater good. People who know that metabolomics is a tool that holds the promise of transforming our society, but who also understand that to deliver on that promise, metabolomics needs to be part of something greater. Projects, networks, infrastructures, they all form around a common goal and make use of the best tools at their disposal. So this year in the podcast, I talk to passionate people who build communities through any strategy, from grassroots movements to decades of networking. Join me to discover the creations of these community builders and how metabolomics is helping them advance their mission. Welcome to the Metabolomist. Today I am joined by Caroline Dricot. Welcome.
Hi, Alice. Thanks for having me.
Thank you for being here. Caroline, you work at the University of Antwerp on a super exciting project that is one of the reasons why I invited you for this episode. But first, I would like to talk a little bit about you and your career so far. So can you tell us a little bit what's your expertise?
Yeah, sure. I would say I'm still very early in my career. Only last month, I successfully defended my PhD. So for me, it feels like I'm just getting started. However, my studies were already 10 years ago. So I studied bioscience engineering with a specialization in cellular and genetic engineering. And I focused on molecular biology. So I was really interested in everything that came down to a few single reactions that could possibly lead to a physiological effect. And also maybe making or inventing a device, living or not, with a specific rationality that could solve a disease. Yeah, so all my studies were actually in Belgium. I already did my bachelor and master thesis at my current lab, so the lab of Professor Sarah Lebeer in Antwerp. And I knew early on that I wanted to do a PhD, but I didn't know it was going to be in the vaginal microbiome field. However, I just stayed at the lab because it was the science was very nice, the working environment was nice, and also, of course, the societal impact of the work, the female empowering, the taboo breaking part. So I applied for a governmental funding, like a personal grant for the FWO, and I got it, luckily. And then my PhD started, and it was about vitamin B2 in the vagina, more specifically when it's being produced by the microbiota, especially the lactobacilli, and how it's involved in mucosal immunology. And then during my PhD, I forced myself to be like a sponge to absorb as many different technologies as I would need to answer my research questions. And that's how I also ended up doing metabolomics at the EMBL at a group of Michael Zimmerman. So yeah, and now I'm a junior postdoc, and I'm trying to find my place again.
Thank you. I think it's really interesting when you start working in a team at a very junior position and you stay. That's really a good sign also that the environment is good. And I could see also, I contacted your PI asking her to be on the podcast, and she immediately said, no, Caroline should do it. She's great. And she really supported you to be on the podcast. So I think it seems like a really nurturing environment. And
yeah, it is.
I think it also speaks to, we will talk in the second half of the episode, we will talk about the ISALA project more specifically. But I think this is also for me what came out of that project is that it's a citizen science project, but that seems really driven by being useful for society, for the population.
Yeah,
and
exactly.
Not everyone gets to do a PhD in this kind of really positive environments on something that seems to have been very inspirational for you. Can you tell us more maybe about the vaginal microbiome? And I'm guilty of this too. Like most people, when they say microbiome, they think gut microbiome. What's special about the vaginal microbiome? What do you think people should know?
First of all, the vaginal microbiome has been neglected for a long time, let's say, and that's mainly because it has a very low alpha diversity, so very low species diversity, and overall, we say the lower the diversity, the better, because the most healthy, most optimal vaginal microbiome composition is when there are a lot of lactobacilli. And these are especially Lactobacillus crispatus, Jensennii, Gasseri, or Inners, but there are, of course, also more diverse vaginal microbiomes. And it's especially this crispatus that has for a long time been associated with health. I'm going to oversimplify it, but we say that the more Lactobacillus crispatus is there, the lower the risk at genital infections, at adverse pregnancy outcomes, but also fertility issues and so on. So yeah, that's like a major thing that you need to understand, that the more lactobacilli, the less diversity, the better, we say.
Can you tell us more, like from the sampling to the analysis, what's similar, what's different?
I don't have a lot of experience with stool samples myself, so I think it's for me it's difficult to say what's different. You have a lot of host DNA when you take a vaginal swab, so that's something to consider. But I think it's also in stool samples the same.
The paper we're going to discuss, it's a preprint because I was too excited for ISALA to finally have metabolomics. I couldn't wait for the paper to be accepted to interview you. But you mentioned you went to the lab of Michael Zimmermann. That was your first experience with metabolomics, I assume?
Exactly, yeah. First of all, I really loved my stay there. The EMBL is wonderful. The team of Michael Zimmermann is very warm. They really welcome you. So I have really positive experiences and memories about that time. What I do think about metabolomics is that it's way more difficult and complex to understand than just genomics or transcriptomics. Especially when you do untargeted metabolomics, you have molecular features, but what is it? You're not sure. You have so many different things you still have to do. You're talking about something, but you don't actually know what it is. So for me, that was very difficult to start working with it. However, I mainly worked there with Denise Legato, and she really explained everything by heart and took me through the steps from putting it on the machine to analyzing it to data quality and so on to interpretation. So that's really nice. Yeah.
Yeah, I think it's great also and I think you need it, to discover metabolomics, you need someone who, you can have someone do the measurement, but you need someone who explains how to make sense of it. So this is the original purpose of this podcast. The very first season was to ask people how they do the interpretation, because this is usually the big bottleneck for people. So I'm happy you had very experienced people who could support you with this, but I guess you had to do a lot of the digging yourself once you had the data.
Indeed. Of course, I had my own PhD, which was very focused about riboflavin and riboflavin intermediates. But then, of course, every PhD person, every postdoc in our lab is also an ambassador for ISALA. So we also contribute to the original project, let's say. So there, the questions were very different. For example, is there a core metabolome for the vagina? And these things they're not really known yet in the field. So it felt very pioneering as well, but also very hard then, of course. But yeah, I think we're one of the biggest problems I feel with metabolomics is still the quantification step, because normally you take a swab, but you're actually never sure how much of a biosample of biofluid you have, especially doing citizen science when people take samples at home and they put it in a buffer directly, then you lose that information. So still there, we're looking for ways to tackle this.
There's no known metabolite you can normalize to, for example. This is done in urine, but this is usually the only matrix where this is done.
Yeah, that's something that we still don't know. So we're trying to figure it out. If there is a metabolite, you can semi-normalize based on DNA or RNA that's there, but it's not enough, let's say. So yeah, we're still working on that.
Yeah, this is one of the big difficulties, especially, and I think it's good if you want to use quantitative metabolomics. There are lots of advantages to that, but of course you need to know what this relates to in terms of amounts, and this can be difficult. Yeah,
I do feel that the field is quite forgiving. They understand that this is still a problem.
And I think this is the most important thing for whatever technology is to understand the limitations and to like put them in front and acknowledge them and then move from there. Yeah. So I think we're clearly moving towards talking about the paper. So let's get right into it. So it's a preprint still, but hopefully soon it will be peer reviewed and published. But so the title is Vitamin B2 Production by Vaginal Lactobacilli Promotes Symbiosis. Yes. So you specified already that your PhD topic was already focusing on these molecules, but can you tell us a bit about what was the starting point for this study? What was your hypothesis?
The really beginning of this story was in a serendipity finding of the ISALA study. The original ISALA project, let's say, was designed to better understand the normal composition, diversity, and function of vaginal lactobacilli. And for this, we wanted to do genomics, metabolomics, and culturing. And during the culturing, so just plating of these vaginal swabs on different agar plates, we noticed that one of these strains, one of these isolates, had this parent green phenotype. And we put it under the UV light and it lit right up. So it was highly green fluorescent and turned out to be due to overproduction of riboflavin. So of course, we directly started thinking about, are lactobacilli perhaps like vaginal fireflies, or like because they're fluorescent? And so we had all these crazy ideas. But then, of course, because we know riboflavin has a lot of beneficial properties in the gut, it's involved in redox, homeostasis, anti-inflammatory reactions, and then also this mate cell activation. We were like wondering whether some of these concepts could also be translated in the vagina. And in addition to this, we know from the ISALA study, and we also cross-validated it for other studies, that Crispatus is often co-occurring, so the most optimal species, let's say, in the vagina, is often co-occurring with other lactobacilli, especially Jansenii and Limotilactobacilli species. And we were wondering whether this could be because of cross-feeding, sharing of metabolites, production of common goods that could either be beneficial between them, but also to the host. And in this hypothesis or this perspective, B vitamins are very interesting. So all of these things together drove us to start looking into the B vitamin levels in the vagina. And then we found indeed that the levels seem to be associated with the abundance of Crispatus.
One question I always have is, why did you choose to include metabolomics in that study? So here it's quite clear because it's like these are the messengers or the molecules that you were literally looking for. That's also why you use targeted metabolomics, I assume. Because one of my questions would have been like, did you start shooting large and then find those ones? But I guess you started directly with this targeted panel of vitamins, or did you also look at other things?
Yeah, we had different research questions. So I'm working with the ISALA data set. So there, of course, we did first untargeted, and then maybe we also did a larger panel of 97 metabolites related to host microbe metabolism of the gut, actually. So we started a bit broader, but then, of course, for me specifically, the riboflavins were the most interesting molecules. For us, still a limitation of this method is the lack of metatranscriptomic data, but it was just the method in which we did the original swabbing in the ISALA study. So the self-sampling, the sending of the sample with postal delivery made it impossible to work with metatranscriptomic data. And therefore, we cross-validated some of our findings, and also with the data set from Jacques Ravel and Michael Franz, which combined metagenomics and metatranscriptomics. And there we also see that in Lactobacillus crispatus dominated communities, so let's say the most optimal microbiome composition, there we see a lot of riboflavin transcripts, so more than in other communities. And we also see that they're coming from crispatus or Jansenii. So all of this together then made us more confident to say, okay, let's go into these functional aspects, these functional experiments.
Yeah. The swab collection and the kit that you sent to the participants, was this designed already to preserve metabolites well? Are the metabolites very stable in these swabs? Because for example, dried blood, we know there are lots of metabolites that get lost, and you just have to know it and work around it. But how is it for swabs?
Again, I think they are how you define stability of a metabolite. It is is important here. We did do some optimization first, and we also said whether, for example, samples that only had like 1 day of transport compared to samples that maybe took 4, 5, 6 days, whether it would have a difference. And there we don't really see a big difference. However, I do think that there will be an impact of, for example, direct sampling, like volatile compounds. Those will be gone either way.
Yeah.
I think for the B vitamins, it was definitely okay. Yeah.
That's great. But it goes back to what we were talking about before also. When you know that your study design requires the sample to be sent in the mail, then you know that you're going to focus on what is left. And that's okay. Like I think there's really nothing wrong with that. Where it becomes more difficult is when people want to stay attached to what was there at the beginning, but is anyway going to be lost in the sample they received. Then, but it sounds like your approach was very rational.
I think so as well. For the follow-up projects, let's say, so we have the original ISALA project, but it's like an umbrella project, we'd say. So we have a lot of follow-up projects as well, and there we try to, if we include metabolomics, to ask people to come to the university to sample there, to minimize like the degradation.
So you could figure out how those vitamins work in your bacterial communities in that study.
I hope at least at some part, yes. So what we currently do not know is whether it promotes, for example, community stability at the population level, because therefore that definitely needs metatranscriptomic data as well as synthetic community experiments, and the field is just not ready for it. We have a paper out on it, but it's really in its infancy. But I focus in this preprint on the host part, and I do feel that there we are some steps further already. So we've seen that, first of all, riboflavin can be transported basolaterally in a 3D model. So let's say that it's actually a model to say that absorption is coming from the luminal side to like the systemic parts. So similar to absorption in the gut, let's say. So it will already be cool to understand whether the in situ production in the vagina could be relevant for the rest of the body. But then on the other hand, we also started looking into whether during this transport riboflavin could affect the redox metabolism of vaginal epithelial cells. And there we do see that some compounds are being increased when they're being exposed to a riboflavin overproducing lactobacilli compared to a low producer lactobacilli. And then also we see some of these aspects of the MR1 mate cell axis that is being activated. So we do see that some of the riboflavin intermediates can induce surface expression of MR1, and this MR1 can then activate the mate cells. But then, of course, it's important to try to understand which signals are also necessary for these mate cells to be really activated in a homeostatic or rather a pathogenic way. And there we do see some signals, but we're still working on this, so we still need to deep dive into this. Then an important methodological aspect that applied in this paper is use of clonal variants. So it's in general, it's very hard to genetically modify clinical isolates of lactobacilli. They are quite refractory to electroporation or anything like that. So this is really a bottleneck. But again, a serendipity finding. So within the same participants where we isolated this overproducer, we found actually in total 8 Lactobacillus strains that were genetically highly similar. There were only a few SNPs, but 2 of them harbored the same SNP in, let's say, the regulatory region of the riboflavin synthesis operon, driving this overproduction. And when we perform untargeted and targeted metabolomics between these strains, we see that there are like a minor of metabolites affected, mainly related to then riboflavin and FAD enzymatic activity. So we use this instead of making a knockout in very hard time-consuming techniques for which you don't have like predicted success, let's say. We use these clonal variants with then, of course, the limitation that there might be some metabolites and some other cell wall structures that might be different, but I think it's safe to say that they're very, very similar.
If I can revert back to one of the things you mentioned. So you mentioned the metabolites going in towards the host and then others coming through. How painful is it when you have to wonder where do these metabolites come from? Because I think, especially in microbiome research, it's the eternal question, does this come from the host or does this come from the microbiome? Yeah. So how have you dealt with that question when you were interpreting your results? And maybe the type of metabolites you're looking at, you can just infer that they come more from a local production. But what were the assumptions you worked with?
I think it's still a very difficult question to answer. In this preprint, we tried to look at whether there were any associations with the questionnaire data of ISALA. So when they took the samples, they also had to fill in an extensive questionnaire of more than an hour on whether they were taking any vitamin supplements, what kind of food they had eaten the last 24 hours, whether they had any vaginal complaints or like activity or sexual activity that was different. So we tried to associate any of the B vitamin levels to these data, but we couldn't find any significant association. Now I have to say that this is not excluding any contribution of the diet, because I do think that there will be, if you take a lot of riboflavin, that some of it will end up in the vagina, and it has to be. Yeah. But however, we wanted to see whether the contribution by the microbiome could at least explain a part of it. And that we do see that there are strong associations with crispatus. So yeah, we cannot fully 100% exclude contribution of the diet or from the host, but finding these riboflases and especially these unstable intermediates, we think that it's more plausible that at least some part is coming from the microbiome, plus finding the in vivo transcription, plus finding that riboflavin biosynthesis is quite conserved among lactobacilli. Yeah. And then often when I get this question, I always think but how do you know it for other microbiome sites? How do you know it for the guts? And there people, especially then in the colon, they are quite willing to understand or to say that it's coming from the microbiome, definitely.
But I think also what you did here and the follow-up experiments you do where you check out the mechanism or you look at how the strains behave ex vivo, that also helps support. So let's say you can make your assumption from what you find in the vaginal microbiome metabolomics and then see if that follows up. I think to some extent you support that with those additional experiments. At least that the bulk of some of the metabolites you look at come from the bacteria rather than from the host. But it's always, this is what I find fascinating, to be honest. It's always this balance you have to keep with all the assumptions you have to make if you want to keep your story going. And there's one question. So I find, just fundamentally speaking, I find the work fascinating, and I think it's super interesting to look at how different species work together or against each other. But of course, we always have to think of the end applications, and also knowledge in the end helps us to make, better medicine, better therapies. From your perspective, what are the end application you expect to come out of this work in general? So when you understand these mechanisms better, where do you hope, maybe is the more interesting question, where do you hope that this will take us?
I would hope that it contributes to the development of new probiotics, specifically for the vagina. However, for example, we have a very interesting overproducing strain that is patented, but it's a difficult road to get a clinical isolate in a real product. So in Europe, at least, it's either considered a medical device or a drug. So it means that it goes through a long regulatory route of clinical trials, let's say, even though the lactobacilli are generally regarded as safe. You can apply them in food supplements and so on. So ideally, I hope we could ever apply one of these riboflavin overproducers as a vaginal probiotic through the application of a cream or a capsule or suppository. But apart from that, I also think that just creating more functional foods or oral probiotic with riboflavin in it or riboflavin overproducer would already help, because there's also this hypothesis in the field that indeed there is a gut-vagina axis. via metabolites, so coming from the gut through the blood to then the vagina, but also via the perineal route of just bacteria. So that would also be a cool application, I think. With then, of course, the benefits of also just an oral probiotic, so perhaps supporting gut homeostasis as well, gut microbiome. Advertising, let's say, a paper of a review of mine. So we have this review of the role of the involvement of riboflavin in women's health and emerging microbiome strategies, where we perform a large analysis on all clinical studies in the past 10 years, linking riboflavin supplementation to women's health. For example, we see that women have increased demand of riboflavin. They need more riboflavin due to mechanisms of menstruation, fertility, but also mental well-being. Or, for example, tissue, so in pelvic organ prolapse. So there, I think that there is a lot of evidence that riboflavin can be very beneficial for women. Hopefully, we can tackle two, how do you say it in English? Two birds with one stone. So both riboflavin and lactobacilli for women's health using riboflavin producing lactobacilli.
Yeah. And my usual last question about the paper is, was there a big challenge for you or something you'd like to highlight? So it doesn't have to be something that was hard and was never solved, but rather something that was really difficult and you found a solution to it. But of course, if there's no solution, you can also put it out there and maybe we get some feedback about it. But what comes to mind?
I think the major struggle we already touched upon is the quantification of the metabolites using the approach that we used. I think we had a hard time of accepting that it's just not really possible in the method that we did. So we're currently looking into ways of solving this, of having really the dry swaps and directly analyzing it, or somehow weighing the swaps up front, but then still there will be variation between swaps. So I don't really have a clear solution, but I think that was for us and is still in the field one of the major challenges for vaginal metabolomics.
Yeah, standardization in general is it's not, I wouldn't say it's a bottleneck, but it's one of the I have the German word coming, the Baustellen, one of the big tasks of the field, to find ways to standardize and to make things more reproducible, and especially when you have large studies like this. So we're going to move now to the community that you're a part of and the ISALA project. For me, it looks like a big community that is growing. I also learned that it's not even limited to Belgium anymore. It's really growing to different countries. So chronologically, can you tell me, because you were there as a young student at the very beginning, so can you tell us a bit, first of all, what the ISALA project is? Because I assume there will be people who listen who don't know exactly what we're talking about. So first that.
The ISALA project originally was designed to better understand the role of lactobacilli in the vagina. And for this, we wanted to do genomics, metabolomics, culturing of vaginal swabs. And an important aspect that we wanted to include, let's say, is moving away from the clinic. Because often people or women were recruited via their gynecologists, but you mostly go to a gynecologist when there is like a problem or you have an issue or whatever. So we wanted to get rid of that bias, and just contact participants. So we had like an entire, at this moment, I was still a job student in lab between my bachelor and my master. So I only observed it from a distance, but there was a lot of effort put in building a interdisciplinary team. So psychologists, people from social sciences, from economics as well, to create a way of presenting this project to people. And then we launched in March 2020, so right in the COVID pandemic, and talking about bugs and microbiome that point was not, but we aimed to get 200 people, and we ended up with more than 6,000 women willing to participate. So we actually had to close all registrations because we didn't have money to analyze all these people. But then we decided to try and get more funding to really engage all these people and include them in a flow 1, let's say, so the cross-sectional study, and then a flow 2 where we would do like the longitudinal study, which we now also included in the riboflavin paper.
I remember seeing the package that you sent to, or that you sent to the participants, and when you talked about the multidisciplinary team, I would almost bet there was a marketer in there, because it was really a beautiful, thoughtful, really well-made thing. Can you explain a bit? I mean, I understand you were very junior at that time, but I'm sure you know what it was made of, and maybe some of the ideas of how to engage people and also what they get back. This is something that's probably very important.
So for the package, we like the idea of Sarah, or she really wanted to look like a small present, like a nice, beautiful female thing, and that's also why we chose for the light pink and the blue to make it feel very girly, let's say. And in the package, there were not only the swabs that we needed, but there was also like an ISALA pin that you could pin on your T-shirt or on your sweater to be part of the community. But then also there were conversation starters in there. So I think 10 questions about some provocative, taboo atmosphere topics. For example, which kind of contraception method do you use? Or have you ever had an infection? Or have you ever had problems with fertility? For women that were participating to talk to their peers, to their family and friends, and hopefully start conversation. Because let's be honest, whoever talks to their mother about these things, I didn't before this. So that was for us very important. And it's really like getting this package was really embedded in the co-creation part of the citizen science. So within citizen science, we actually have 4 levels, which are gradually requiring more contribution of the participants. So at first you have minimal involvement. Next you have contribution. The third level is collaboration. The fourth is co-creation. And there the citizens are really also like scientists, let's say. So we really asked them also what we should focus on. For example, we welcomed every thought that they had during the sampling, or ideas for follow-up studies, for example, or ways how they would like to have the results communicated to them. So we really focused also on social media and having everything very easy to follow, with blog posts, with social Instagram stories, updates this way. So in this way, we've tried to really build a community of followers, let's say, as well, which we can now engage also for other projects. So it's really cool to keep
Now, seen from the outside, I saw this project a few years ago. I think Sarah was giving a talk somewhere, and that was the first time I heard about it. And then I looked into it, and it really, over time, it really looks like it's not just a scientific project. It's a movement. Like, you really built that community, and the use of social media. Of course, scientists now are encouraged to do this, but I think this project really did that really well, and not in a manipulative way, but in a caring way. This is how it transpires for me. Like, really to be in this together with the participants. And I know for large cohorts, so much larger cohorts than this, a question that often comes up and that can be a bit complex is, what results do the participants actually get? So beyond all the goodies and the networking and the community feeling. So what do you actually share of the results with the participants?
So first of all, we try to share the results as soon as possible. Let's say as soon as we're confident about them, we communicate them to the participants, and they get an email, and they have like a password and like a username and a password, so they can enter to their own personal profile. And so we communicate their microbiome composition. So every person gets like a pie chart with their percentages of species found there, as well as a clear explanation of what that species does. So in layman terms, we try to do that. For example, Crispatus is the most optimal one because of this, this, and this. But then also when you have a less optimal one, we also try to mention it, but in a very cautious way, because we don't want to worry them. We don't want to create problems where they're not.
And I guess like today there are no clear diagnostic consequences that are taken from those results, or like there's no alarming diagnosis that would come from this yet. Maybe in the future.
We always say that if you don't experience any problems, there's nothing wrong. Also, either when you have a more diverse profile, that's fine. But what we did do was then include contact information of a general practitioner that was involved in the study. So if they had any questions or worries, they could always ask them. So I think that's one of the main things. And then with follow-up studies, we also try to include more and more. We also update them when we have a paper out, and we try to explain it in a less scientific way, let's say, or more easy to understand way. And we also organize a lot of events, or we contribute to events for kids, for adults. We have in Belgium this thing, it's called Nerdland Festival. So it's like a very big festival of an entire weekend, where we once had a stand where we interacted with a lot of people. So we really try to bring it back to the people as well.
It's really, really a great initiative. I love it. So it's been now running for more or less 6 years from the very beginning, right?
Yeah.
So what does the future look like for ISALA?
It always depends on funding, right? No, we're growing. So as you mentioned, we have these other sisterhoods, like many countries are developing their own ISALA-like project, or we give them a framework of how we did it, let's say. But all of these countries, all of these projects have their own ownership, their own way, and also in every country there are other cultural aspects that you need to take into account, or that you want to focus on in your project. So we are still working on this, and then also we have to move from observation to more mechanistic studies, like we did in a preprint, and there are a lot of follow-up studies. For example, Luna, a project that's looking at influence of menstrual hygiene products on the vaginal microbiome. So, for example, it would be ideal to have in some kind of way guidelines to communicate to the public, to really have that mechanistic proof evidence that would help a lot of women to maintain or improve their vaginal health. So how that will look like, I'm not sure yet. I hope I can still be part of it. But yeah, that would be, I think, the ideal goal, to really like make an impact
because there are still a lot of things that remain to be known, I guess. So I guess, for example, when you have the sisterhoods in different countries, eventually you want to compare the healthy microbiomes, because I know for gut microbiome it's radically different between continents, between countries. Do you already know, just from literature or even from first results, if there are these? Because for the gut we know it's a lot based on diet, the differences, but like, do you know if there are such radical differences for vaginal microbiome between countries that have been described?
There is. So indeed, in general, let's say world population wide, Lactobacillus crispatus is the most beneficial one, but we do see that in a lot of Hispanic populations, for example, much more diverse profiles are very, very common. So we work a lot together with Peru, because the ambassador of, like, the manager of the sisterhood, Sandra Condori, she's from Peru, and that's how we initiated this link. And there we do see that there are a lot of changes or differences in having more diverse profiles with also healthy women. But for example, then also there, the most common used menstrual hygiene product is a menstrual pad, for example. So to understand then how these activities or these lifestyle habits would affect it. Yeah, it's hard to already disentangle from diet, for example, because there are a lot of differences between populations, but to really already pinpoint which is the one most contributing factor, it's difficult.
Mm-hmm. Yeah, but I think this is one of for me, the very interesting things, because there's so much that comes from how we live our lives, how we treat our bodies. And I guess, I mean, you mentioned it a little bit at the beginning, but like for the gut microbiome, it's now clear that there are very strong ties to health. Like when your gut microbiome is imbalanced, this usually associates with many diseases. Is there something like that for the vaginal microbiome that is known already?
What do you
so
mean
that
specifically?
you'd have an unusual vaginal microbiome for the population you come from, and that would associate with people who have, for example, chronic disease. Is there something known about this, or it's too soon to tell?
I think it's too soon. Also, for us, we're mainly looking at a healthy population.
Yeah.
So I mean, in ISALA, it's self-proclaimed healthy, and we took into account whether they were taking any drugs or they suffered from a disorder or whatever. But for most studies, we try to exclude those participants to not have that bias. But I do think so. I do think that especially also immune disease or chronic diseases linked to immune response, that there will be differences.
Yeah, but once you have a good picture of the healthy microbiome, then I think that would also be a nice next step to move towards different types of disease and see how that changes. So many exciting things for this project. I really look forward to following ISALA. Thank you for introducing it to us. So the last question, as always, is what's your favorite metabolite and why? I have a feeling I'm going to easily guess which one it is, but so... Which metabolite did you choose?
It's an easy choice. It's riboflavin. Just because, it's fluorescent,
I think it's beautiful how you focused your attention on it, like how you had the idea to look at it and to look further into it.
No, I think it's such a versatile compound. It's involved in so many different aspects, which makes it very hard to study. We mentioned micro posts. Then in humans, there can be so many different other things, but I think, it's a common good. It's one of those molecules that's good for you. So I think, that's my favorite one.
Beautiful. Thank you very much, Caroline, for taking part in the podcast and presenting your work and this beautiful project. And I wish you all the best in the future.
Thank you. Thank you for having me and giving me this opportunity, Alice.
Thank you for listening to this episode of The Metabolomist, where we explored one of the many communities that leverage metabolomics for the greater good. To make sure that you don't miss out on our next episode, you can follow the podcast on your podcast app or go to themetabolomist.com and register to our newsletter. Until then, I wish you to get to use metabolomics with the same passion and the same dedication as my guest today, and don't hesitate to reach out to me on LinkedIn at Alice Limonciel with your thoughts on today's episode.